Welcome to the video that is all about the history of microbiology. And while there are many, many people and experiments that led to our modern day understanding of microbiology, for the point of this particular video, and being that we're only in Module 1, Unit 1 of the course, we're going to do big picture overview, and I will introduce you to other key people later in the course. So, what we're going to focus on initially is the spontaneous generation debate. Debate is letting you know that not everybody agreed with this theory of spontaneous generation at all times. So, we're talking hundreds of years ago, right? This whole conversation starts in the late 1660s, early 1670s. So around that time and before then, people often thought that, sickness or disease was just a consequence of poor life decisions. Perhaps they are being punished by some higher power. They thought that certain, animals, for instance, that have microscopic eggs or larvae that we can't see, right? They just thought that those animals just boop out of nowhere, just magically, spontaneously generated, right? They literally just came out of nowhere. And this, makes sense that it was a common theory because we didn't have microscopes and tools that would allow them to visualize disease-causing bacteria or the microscopic parts of an organism's life cycle. And so but people were suspicious that maybe there's more to the world than what we can see. Okay, so I'm going to start the conversation with Anton van Leeuwenhoek, even though technically he comes a few years after the first big experiment about it, um, because I want to kind of keep all the experimental data-driven stuff together. So I want to introduce Anton van Leeuwenhoek first. And what he did was pretty snazzy. Okay, he created his own homemade makeshift microscope, and he used it to observe and describe different living organisms that he found. And so what he did is he made this homemade microscope, and then he went out and collected samples like from everywhere, right? Pond water, soil. He scraped his teeth. Okay, and then he would use his microscope to see what there was, and then he would create detailed drawings and notes in these journals. And I think those still exist somewhere. I want to say the Pasteur Institute in Paris, but I might be wrong. But just these very extensive notes on what he observed. And he called these tiny organisms animalcules. So, cule like a molecule, and then animal. Perhaps they had more animal-like appearance to him and were teeny tiny, hence the molecule reference. But essentially, Van Leeuwenhoek really provided some of the earliest evidence that this invisible microbial world existed. And this really laid the foundation for the field of microbiology. So it's really, really important. One thing I forgot to mention, y'all, is you do not need to memorize these dates. You do not need to memorize the first names of these scientists. I will just say Van Leeuwenhoek. Um, what you will need to know is the last name and then kind of what they did and what that meant for the field. Okay, so he made a microscope, observed specimens from different areas, and documented what he found, and he referred to them as animalcules. All right, so let's start looking at kind of some experimental setups that were done um, that really kind of got this spontaneous generation debate going. So, Reddy, what he did is he placed raw meat in an open jar, a cork cork covered jar, I should say, and then a gauze covered container. Sorry, y'all. I don't know why my speech is against me. And so you can see the picture of this setup right here. And what he found is that maggots developed only where flies could access the meat. And this supported the idea that maggots came from flies rather than spontaneous generation. Okay, so remember I mentioned that some organisms, part of their life cycle, they're really small. Technically, fly eggs are not microscopic. They're only about a millimeter. And so between their size and their color, they can be really, really hard to see or even confused as part of the meat. And so people tended to think that maggots just spontaneously generated. And so when Reddy only saw that there were maggots associated with the open containered or meat that was in the open container, he said, "See, guys, they're not coming from nowhere, right? Because if that was the case, if maggots spontaneously generated, we would see them in the cork sealed container and the gauze covered container." Now, a lot of people are resistant to change, right? They aren't just going to abandon their beliefs at the drop of a dime. And so, what do you think some pushback was on this experimental setup and the findings? One big one was the lack of oxygen in the cork sealed container, and perhaps just drastically reduced oxygen in the gauze covered container, right? Because some critics would say, right, that these maggots are large and they're complex, right? They're eukaryotic. And so they must need oxygen like all living things do. Because remember, at that time, they didn't know any better. Okay. So while we know today that Francesco Redi was very much heading in the right direction with his thoughts, people weren't quite ready to be convinced. Okay. So let's now look at John Needham. And Needham is responsible for kind of rekindling this idea of spontaneous generation. So what he did is he got some broth, I think it was like some meat broth and some plant broth, and he briefly boiled that broth, kind of let it cool for a little bit, and then he sealed the flasks with cork. And what he saw was that there was still microbial growth. And Needham said, "See, guys, this shows you that these microorganisms can arise spontaneously from non-living material." Right? He said, "I boiled this. That should have killed everything. And look, there's still stuff growing in here. Spontaneous generation is a thing." Okay. So we nowadays know that he's wrong. So what do you think the flaw was in his experimental setup? Remember, he put some broth in a flask, briefly heated it, let it cool, corked it, saw growth. So there's a couple of things. So really this heating process that he was boasting about was not sufficient. Okay, it likely wasn't hot enough or long enough. And if he let it cool before corking it, then that would also have introduced, right, any microbes from the air. So we now know that even though he tried really hard, Needham's setup was flawed. Okay. And so it took a hot minute, right? If you do look at the timeline, about 50 years later, Lazzaro Spallanzani essentially redid Needham's experiment, but he tried to correct for those flaws. So what he did is he took flasks full of broth. He boiled that broth longer, and then he immediately sealed the flasks. And he sealed them a bit more effectively. Cork isn't always, uh, like non-penetrable, right? Air can get through, not a lot, but it can. And so he sealed this flask more effectively. And what was found is that the broth remained sterile unless the flask was open. And so, what this proves is that the microorganisms must be coming from other microorganisms in the environment, even if we can't see it. Okay? Because after he boiled and sealed it, nothing grew, as to be expected, because anything that had been alive in that broth was killed. Okay? And so, if spontaneous generation was a thing, you would have still seen growth, right? Because they would have just grown once things cooled down. But that did not happen. But people still did not want to abandon the spontaneous generation ship. Okay. And so, what do you think their big pushback was? Once again, it was about oxygen, right? They said that air is this vital force. And by sealing off that flask, we're not letting this vital force of air into the system. So, of course, things can't spontaneously generate. Okay. So, up until this point, we didn't really have a good solution for showing that. So, later, again, almost half a century later, here comes Rudolf Virchow. And while he didn't, conduct some mind-blowing experiment or anything, what he did help do is establish this principle and now theory of biogenesis. So if you look at the word, right, bio and genesis, it's basically saying that cells are arising from pre-existing cells, right? Living things generate other living things. Okay. Because if you think about it, it still makes some sense, right? That people didn't want to abandon the idea of spontaneous generation because they had nothing else to grab hold of, right? There was no real alternative theory that could explain what was happening. And so when Virchow says, hey guys, maybe cells come from pre-existing cells, maybe microscopic things come from other microscopic things. And so this now gave people a theory to hold on to and to compare, data to. Okay, look, they can look at all this pre-existing data and even future data and say, hey, does that fit within this theory? Does it support this theory? So this was a big deal. And not too long later, Pasteur said, all right, y'all, hold my flask. I am going to shut down the spontaneous generation debate once and for all. Okay. And how he did it is through the use of these S-shaped or swan-necked flasks. Okay. So I always refer to them as an S-shaped flask because you can see the S shape here. Okay. And so what happens is he had the flasks, he put broth in it. Okay. He boiled it just like before, right? This sounds strikingly similar to what Needham and Spallanzani did. But the difference is this S-necked flask, because what you can kind of see here, if you look closely, is there's air. Okay, so this end is open. So what that means is air can get in to the flask, okay, but nothing can travel all the way down this flask to contaminate the broth. Okay. So even if dust, or as we now know, microbes are able to like fall into this opening, they can't, fall down and climb up this flask to get into the broth. Okay. So that's the beauty in this design. Okay. The broth is unreachable, but air is still getting into the system. Okay. So again, he put the broth in this S-necked flask, boiled it, and lo and behold, the broth remained sterile, even with oxygen being able to get in. So this was extremely strong evidence that microorganisms arise from existing microorganisms and that spontaneous generation is not a thing. Okay? Now, Pasteur actually took it one step farther. Okay. And so what you see in the top portion of this image is that, he boiled the flask, oxygen, dust particles, bacteria, they got in, they could never reach the flask and it remained sterile for a very, 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 very long time. I think some of these are also still available in the Pasteur Institute. But what he also did to take it a step further is for some of the flasks, he broke them, as you can see here in this bottom middle picture. And then he just left it to sit out. And what do you know? It eventually had growth in it because microbes from the air were now able to get into this open flask and grow. So he showed two things. He showed that um, spontaneous generation was not true, and therefore biogenesis is the more likely theory. But then the second really big thing that he showed is that there are microorganisms in the air. Because how else did this flask get contaminated? Pretty snazzy, eh? All right. We're going to wrap up this part of the video with a little bit more information on Pasteur and then just a little bit of information about Robert Koch and Edward Jenner. So, beyond spontaneous generation, Pasteur did a couple of things. He was able to demonstrate that microorganisms are responsible for fermentation. And he was able to show that certain microbes, okay, are able to essentially cause specific changes in food and beverages. And we're going to talk about this more in Unit 2. But this finding led to the development of pasteurization. You can see his name here in the name of the process. And pasteurization is basically using controlled heat in order to minimize the number of microbes, associated with milk, for instance. And this is going to reduce spoilage and, disease-causing organisms to live in there. He also did some work that helped establish germ theory, which basically is linking microbes to disease. Okay, right? Not poor life choices, but some microbe is actually causing disease. Um, and it's really pretty snazzy. And part of that debate is actually the next guy, Robert Koch. Him and Pasteur were kind of in this battle to try to prove germ theory. And it turns out Coke won, which was pretty cool. Um, and we'll talk a little bit about it more in lab, but he was able to win because somebody in Coke's lab suggested using agar. And so they were able to develop agar plates that could easily be inoculated and incubated and grow organisms. And that actually helped give him the edge over Pasteur. But essentially, Robert Koch did provide experimental evidence that connected a specific microorganism with a specific disease. And so he developed Koch's postulates, which is essentially this kind of systematic framework or checklist kind of that allows you to determine whether or not a specific microorganism or microbe, now that we know more about viruses, causes a particular disease. So this helped move germ theory from just this general idea to something scientists could actually experimentally test. Now, there are some flaws in Koch's postulates, right? Nothing's perfect, but this really laid the groundwork for people to associate and link a microbe with a disease. Lastly, Jenner is somebody that we'll talk about a little bit more in Unit 3, and he observed that exposure to a relatively mild disease like cowpox protected people from smallpox, which is a much more dangerous disease. We'll dive into how he did this, not exactly the most ethical work, but his work did demonstrate the basic idea, right, that we can expose ourselves to a less harmful related pathogen and give us protection against a future more scary disease. So this really laid an important foundation for modern vaccination and immunology. All right. So I know this was a lot of information, y'all, but as you're working through this module and you're working through this part of the module, maybe make flashcards that has, Reddy's name on the front. You kind of just spell out what he did and what that meant for the field. And to really make sure you can explain to me or someone else what each person did and the impact that they had. All right, y'all. Please let me know if you have any questions. I am always here to help. Bye, y'all.